Welcome to New Botany, and I'm following up my interest in willow herbs by doing a more scientific analysis on them. Now, most of these common species you find around cities are known as Epilobium ciliatum, but I think there are two kinds that might be different. This one with white flowers which change to pink, which is known as pseudorubescens, and the purely pink flowered one called adenocaulon, which set seed a little bit earlier than the white flowered form. I think there's evidence that these two different kinds of ciliatum are different, but the DNA analysis proves otherwise. So I've chosen another method of analysing these plants using paper chromatography of acetone extracts. And if you run these samples on chromatography paper, you can see visible substances, and there are also invisible substances which could be used to identify different species. Now, this is a UV light illuminated chromatography paper, and you can see that the UV light illuminates or visualizes other bands that you can't see with the naked eye, and it also enhances visible light absorbers as well, so that you can see extra bands. These are the low short distance running bands. This is Epilobium hirsutum, a much more obviously different plant in the Epilobium group. It's got a very large flower, and if you run the paper chromatogram on this one, Epilobium hirsutum shows an extra band at 0.69 reacting with UV light. So these high reacting bands with UV light may be a means of identifying or characterizing different species. Here we've got a very small plant which has the same kind of flower as Epilobium montanum, though it may well be a hybrid. This is one that's growing in my garden. It's different because it's much smaller, and the flower seems to have malformed stigma lobes, which are more like Epilobium adenocaulon. Now, if you run the paper chromatogram of this plant, you can see that the UV illumination only shows one band. These UV light absorbing bands are known as flavonoids because they have high mobility in this particular solvent. You calculate RF values for each of the substances that runs in a paper chromatogram, and looking at the UV absorbing bands, there's one at 0.6 to 0.7 and one at 0.7 to 0.8. All the adenocaulon and pseudorubescens and hirsutum samples showed the same bands, but montanum and the montanum hybrid showed a lack of a band at 0.6 to 0.7 RF value. So I have been able to detect differences. I wasn't able to show a difference between adenocaulon and pseudorubescens, which I was hoping to be able to do. But there we go, you can't get everything. It may be possible to show the differences between these plants in other ways. So here are my conclusions from the experiment, and that's a nice picture of pseudorubescens there. Polyphenol chromatography shows some differences between Epilobium species, and that is an interesting finding. They're not all the same. Epilobium montanum and its forms only possess one high mobility flavonoid band at 0.7 to 0.8 RF value. It's not possible to resolve Epilobium pseudorubescens and adenocaulon, as they have identical polyphenol bands. So please like and subscribe. Thanks for watching, and I do have a PS postscript here with Epilobium tetragonum results, and this plant, like montanum, it also lacks the RF 0.6 to 0.7 polyphenol band. And here are the pictures here. You can see the polyphenol band at the top there, on the top right. There is only one. Thanks for watching.